Hello, my name is Noah Okada, and today I'll be presenting the work I conducted under the guidance of my mentor, Dr. Mercedes Balcells-Kemps at the MIT Institute for Medical Engineering and Sciences. Our work sought to understand the effects of stress and inflammation on the brain microvascular endothelium by creating an in vitro model of the blood-brain barrier. To give a brief outline of the presentation, first, I will go over some of the background information about the blood-brain barrier and the reason it is important for us to develop models to better understand the effects of stress and inflammation on this barrier. Next, I'll provide an overview of the methods we use to develop our model of the blood-brain barrier and the various experiments we developed to simulate inflammation within the cerebral vascular system. Then I will discuss my preliminary findings based on the analysis of protein expression by the endothelial cells. And finally, I will conclude by discussing the implications of this work. So the question we're all asking is, what is the blood-brain barrier? And the blood-brain barrier, simply put, is a selectively permeable barrier that separates your brain from the rest of your bloodstream. And this is vital for maintaining your health as it protects sensitive neuronal tissues from pathogens and neurotoxic chemicals that may exist within your bloodstream. Because of this vital role in maintaining your health, dysfunctions of the blood-brain barrier often results in disease. And there is a growing amount of medical evidence that correlates dysfunction of the blood-brain barrier with the incidence of stroke, Alzheimer's disease, dementia, and other neurodegenerative diseases. And that is why this highly sensitive neurovascular unit is important to study and understand so that we can diagnose and prevent diseases of the brain. In order to study this, it is important to understand the structure and components that exist within this unit. As you can see in the image, the blood-brain barrier is composed of various cells such as astrocytes, parasites, and endothelial cells. But the primary barrier that separates the central nervous system from the bloodstream is the endothelium. The endothelium is composed of endothelial cells. These cells join together to form what is known as tight junctions. Tight junctions serve to tightly regulate the entry and exit of molecules through the blood-brain barrier. And disruptions of these tight junctions lead to dysfunctions of the blood-brain barrier. While there are many proteins that make up tight junctions, in our experiments, we studied two key proteins, claudin 5 and ZO1. Tracking the expression of these proteins helped us to quantify the existence of tight junction between cells. Now, looking specifically at inflammation, studies have shown that hypoxia, a condition when tissues are deprived of the proper oxygen they need, can lead to the production of inflammatory cytokines, which can downregulate tight junction proteins. And based on these previous findings, our research sought to ask the question, how does inflammation impact the expression of tight junction markers? Based on the background information, we hypothesized that there would be a significant difference in the tight junction protein expression between cells cultured in hypoxic conditions and those cultured with pro-inflammatory cytokines. To test these hypotheses, we developed a model of the blood-brain barrier by growing human brain microvascular endothelial cells in the lab. Over the course of our experiments, we studied three different groups of endothelial cells. The first group was cultured in the presence of cobalt chloride, a compound that has been shown to model the effects of hypoxia by altering the cell's ability to uptake oxygen. Our second group of cells was cultured in the presence of tumor necrosis factor alpha. This is a common pro-inflammatory marker that is used in the immune system to signal inflammation. Finally, our third group of cells was a control condition and was grown in optimum conditions for endothelial cells. We grew these cells over several days and then employed various techniques to measure the expression of the protein marker ZO1 and claudin 5. The figure here displays the initial results from our hypoxia experiments. In this experiment, we studied the effect of differing concentrations of cobalt chloride on the cells. As you can see from the images, each concentration displays a different degree of fluorescence. These fluorescent images display the expression of proteins ZO1 in green and claudin 5 in red. Our findings showed that as you increase the cobalt chloride concentrations, the ZO1 expression decreased, which suggested that hypoxia negatively affects the tight junctions and can disrupt the blood-brain barrier. Building off of these findings, we compared the protein expression for cells grown with TNF-alpha, cobalt chloride, and the control. Our results showed that although TNF-alpha impacted the expression of proteins, the effect of cobalt chloride on the viability and expression of cells was more significant. Namely, there was a reduced expression of Claudin-5 in cells cultured in hypoxic conditions. While these results are preliminary, they provide pivotal information that will help us to understand the effects of inflammation and stress on the blood-brain barrier. The development of this work comes at a crucial time, as cerebral vascular disease remains the 5th leading cause of death in the United States. Furthermore, in the past 2 decades, the deaths from Alzheimer's disease has increased 145%, and this trend does not get better, as patients who have suffered stroke are 4 times more likely to develop vascular dementia. These statistics point to the severity of the issues that can arise when the blood-brain barrier is disrupted. Although these statistics are daunting, it is the continued development of models such as these that will help researchers and clinicians to better understand and treat disease. And it is my hope that this research will provide relevant findings that will guide the work of future research as we seek to develop solutions in the field of biomedical engineering. I would like to thank you for listening to this presentation and acknowledge the hardworking mentors and peers who have helped develop this project. I'd like to thank Dr. Balcells-Kamps for being my research mentor during this project, Gemma Mullins for her supervision of this research, and Hector Cantubueno for being my lab partner and working alongside me at the bench. Thank you.